Let's take a look at blood type now. Maybe you know your blood type, and if not, you'll have a chance to determine your blood type in class. So blood type is genetic. And remember, genes code for proteins. So you have proteins on the surface of your red blood cells called antigens. Lots of cells have antigens. If you've taken microbiology, you learned about them in there. When we study the immune system, we're going to learn about antigens. So it's not just red blood cells that have antigens. But the ones on your red blood cells are what determine your blood type. And if you remember from chapter 2, proteins are all about shape. So these antigens come in different shapes. And your blood type is which shape antigens you have on your red blood cells. So there are 2 different genes that give you antigens on your red blood cells. There is the ABO, and there is your Rh positive or negative. So for ABO, you can have the A gene will give you the A antigen. There is the B gene, codes for the B antigen. And the O gene codes for no antigen. So O just means you don't have an antigen. There is no such thing as an O antigen. O just means you don't have it. And for Rh, if you have capital D, that codes for having the D antigen. Or you can have lowercase d, which like O means no D antigen. We're going to fill out this chart, and as we fill out this chart, you'll start to see the pattern and it'll start making a lot of sense. So first for the genetics, it's 2 genes. So you have the ABO and you have the Rh. Let's say you either have AA, means each parent gave you an A allele, or you could have AO. One parent gave you an A and one parent gave you an O. If that is the case, you will have A antigens on your red blood cells. So A is dominant, O is recessive. If you have 2 capital Ds, so each parent gave you a capital D, or you have one big, one little, then you would also have the D antigen. If you have A and D antigens, your blood type is A positive. Let's say you have, again, either AA or AO, but this time each of your parents gave you a lowercase d. In this situation, you would have the A antigen, but because both of your D's are the lowercase recessive ones, you would not have a D antigen. So all you have is A's. This is A negative. Let's do the same thing with B's. If you have 2 B's or a B and an O, you get B antigens. B is dominant over O. If you have two capital Ds, or if you're heterozygote, you get the D. This is B positive. If you get a BB or a BO and then two lowercase d's, you just have Bs, and this is B negative. Okay, are you starting to see the pattern here? Let's suppose one parent gives you an A and the other parent gives you a B. Well, A and B are codominant. So you would make both types of antigens. If you have both the A allele and the B allele, you'll have both the A and B antigens. Let's say your parents give you at least one big D, then you would have D. So you would have all three antigens. This is AB positive. If one parent gives you an A and the other a B, and they each give you a lowercase d, you have A's and B's, but no D's, so you're AB negative. O is the recessive trait. So in order to have type O blood, you have to get an O allele from each parent. And remember, there's no O antigen. That just means you don't have A or B. If you have at least one big D, you would have D's. This is O positive. And if each parent gives you an O and each parent gives you a lowercase d, you have no antigens at all. Just this naked red blood cell. This is O negative. So that's how you determine the blood type. It's based on which antigens you have, which are coded for by your genes. Now we can look at antibodies. Antibodies are made by your immune system against foreign antigens. So antibodies are proteins. And again, proteins are all about shape. They are shaped to fit to an antigen. So you can think of antibodies and antigens are like a lock and key. So remember, you're going to have antibodies against whatever antigens are foreign to you. If you have A positive blood, that means you have A and D antigens. You will therefore have B antibodies because B is foreign to you. If you're A negative, you have A antigen. You do not have B or D. So B and D are foreign to you, you'll make B and D antibodies. If you're B positive, then you do not have A antigens. They are foreign to you, so you make A antibodies. Your immune system doesn't like any antigens that are not naturally a part of your body. It makes antibodies to attack them. B negative has A and D antibodies. AB positive, notice if you're AB positive, you have all three, A, B, and D. Nothing is foreign. AB positive people do not have any of these antibodies. AB negative has D antibodies. O positive has A and B antibodies. O negative, because they don't have any antigens at all three of them are foreign. So they would have all three antibodies. Antibodies are important when it comes to doing transfusions. So transfusion, when you can put blood from one person to another, and a lot of times you need to do this in order to save somebody's life. This is the antibody. These are the antigen. In this case, notice the antibody does not match the antigen. So here, see this is a triangle shape, and these are round. So in the top one here, we have no match. This lock and this key do not go together. The antibody does not fit to the antigen. So the antibody leaves the blood cells alone. So we could say that this is an A antigen and that the antibody is a B antibody. They don't fit together, so they leave each other alone and everything's fine. That's what you want. You want your antibodies leaving your cells alone. Down here though, notice we do have a match. Their shapes fit. So if this is the A antigen, and this is an A antibody, they fit together. And notice that an antibody has two binding sites here. So it will bind to two red blood cells. So one antibody binds to two red blood cells. And this goes around. So if you had a red blood cell here, you had an antibody binding to it, and another one here. Well, then you could have another antibody here binding, and another blood cell here. And you could have an antibody here, and a blood cell here. And it could keep going. And what you end up with is a big clump of red blood cells. And this clump is called agglutination. Now, it's important not to mix up agglutination and coagulation. Students tend to want to do that. Agglutination is when an antibody binds to red blood cells and causes them to form a clump. Coagulation is when you get a cut and you make a blood clot so you stop bleeding. Different things. Don't mix them up. So when you're doing blood transfusions, you want to avoid agglutination. Agglutination is deadly. If those blood cells are all clumped up, they're going to clog your blood vessels, the blood's going to stop flowing, and you will die. So you don't want this. So when it comes to doing blood transfusions, you have to avoid agglutination. So let's look at which things can be transfused. Let's say you have type A positive blood. And you need a transfusion. We want to know what kind of blood we can put into you. If you have A positive, that means that you have B antibodies. Well, if you have B antibodies, that means you will attack and agglutinate B antigens. Agglutination is a bad thing. You don't want it. So if you will agglutinate B antigens, that means we cannot put any B antigens into you. So you must avoid B antigens. So let's look through the list and see what you can get. So we'll start off with A positive. There's no B antigens on that blood cell. So A positive is good. You can always get your own blood type. If we look at A negative, again, no B antigens there. so you can get A negative. If we look at B positive, well, it has B antigens, so that one's out. B negative has B antigens, that one's out. ABs, the AB positive and AB negative, both have B antigens, so those are out. The O's, neither one have any B antigens, so those are both good. So if you have A positive blood, there's a list of blood types you can receive in a transfusion. Let's do another example. Let's do B negative now. If you have B negative blood and you need a transfusion, this means that you have A and D antibodies. Look at your chart, look at the B negative. B negative has B antigens. A and D are foreign, so you have A and D antibodies. So this means that you have to avoid A and D antigens because you will cause agglutination if you get an A or a D antigen. So let's go through our list. A positive, well, it has both of them, A's and D's, so you can get lots of agglutination. A negative, it has A's, you'll get agglutination. B positive, it has D's, you would get agglutination. B negative, yes, no A's, no D's there. Okay, AB positive, again, you would agglutinate with two of those, the A and the D. AB negative, your antibodies would cause agglutination with the A. O positive has a D. You would cause agglutination. O negative has nothing there. You can get O negative. So if you have B negative blood and you need a transfusion, you can get B negative or O negative. Okay, some of you might be thinking about universal donor. You may have heard of that. O negative is the universal donor. You can put it in anybody. Every person can receive O negative blood. The reason for that is they have no antigens. Antibodies bind to antigens. Well, if you have no antigens, there's nothing for an antibody to bind to. So it doesn't matter what kind of antibody the recipient has, they can't bind to a blood cell if there's no antigens on it. So antibodies cannot bind. So O negative blood will not agglutinate. There's nothing for the antibodies to stick to cause agglutination. There's also a blood type here that is the universal recipient. These people can receive blood from anyone else. So if you have this blood type and you need blood transfusion, you can get blood from anybody. That one is the AB positive. AB positive is the universal recipient. They have no antibodies. They have nothing to cause agglutination. Nothing is foreign to them. So again, this blood is not going to agglutinate because they don't have any antibodies. You can put anything in an AB positive person and they're going to be fine. There's another situation that comes up with blood types that has to do with pregnant women. There are circumstances in which the fetus can get hemolytic disease. And the good news is that today with modern medicine, we have treatments for this. But in the old days, it would cause miscarriages and stillbirths before we had treatment for it. So hemolytic disease occurs only under certain circumstances. The woman has to have negative blood type. And she has to have a child with positive blood type. So that's the only time it's an issue. So only women with negative blood type need to worry about hemolytic disease. And the child can inherit positive from the father. So if you have a woman with negative blood type and she marries a man with positive blood type, she'll have to get the RhoGAM shot in order to protect her children from hemolytic disease. So what happens here? We're dealing with positive and negative, so we're dealing with the D antigen, and antibody. Okay, if we look at our pictures here, in the first picture, this woman is pregnant with her first child. This is her first pregnancy. So here we have a negative woman. This is her first pregnancy, and she's carrying a positive fetus. During pregnancy, mother and fetal blood don't mix. The placenta separates them. But during birth, there is some mixing. Fetal blood will leak into the woman. So remember, this fetal blood is positive. It has D's on it. And this leaks into the mom, who is negative. This sensitizes her. This sets off her immune system. Her immune system makes a bunch of D antibodies because this positive blood is foreign to her and her immune system doesn't like it. So it sensitizes her. She will make lots of D antibodies. This happens while she's giving birth, though. So even though she's making D antibodies, they can't attack her child because this child's on his way out anyway. So this child is fine because he is out before the mom's immune system can actually do anything. So women who have negative blood type can always have the first child. That will be fine. The issue comes with the second child. So now here you can see between pregnancies, here's all those D antibodies. She built up a bunch of them. She's now sensitized. Now we have her second pregnancy. And if the fetus is again positive, now those D antibodies, you can see, are crossing the placenta and attacking the fetal blood. Now, this isn't an issue with A and B. A and B aren't going to cross the placenta and attack the fetus. It's only the D's that do this. So now this fetus gets hemolytic disease. And this is fatal. So this will cause miscarriage or stillbirth of the fetus. So in the old days, before we had medical treatment, if you had a woman who was negative, married a man who was positive, they could have one child. After that, she would probably have miscarriages or stillbirths, unless if she had a negative child. So this woman could have a negative child and would be just fine. It's just a negative woman having a positive child that's an issue. So today we do have medical treatment. And that medical treatment is RhoGAM. So now anytime a woman who has a negative blood type gets pregnant, they give her a RhoGAM shot. So a pregnant negative woman gets RhoGAM during the pregnancy and again when she gives birth. What the RhoGAM does is it travels through the mom and into the fetus. So they give her the shot while she's pregnant. The RhoGAM comes in and it coats the fetal blood cells. So the RhoGAM goes to the fetus. And it coats the fetal blood cells. It's like camouflage. Then when the woman gives birth and that fetal blood enters her body, these cells are camouflaged. They're coated in RhoGAM. Her immune system can't see the D antibodies. It doesn't see the D antigens. It doesn't detect that they are there. So because the RhoGAM is camouflaging those fetal red blood cells, when she gives birth and the fetal blood enters the mom, her immune system doesn't know. They are hidden from her immune system. So she does not become sensitized. She does not make the D antibodies. So this, if the woman gets the RhoGAM shot, that would not happen. There would not be those D antibodies there. Then when she gets her second pregnancy, there's no D antibodies to go attack that next fetus. So the point of the RhoGAM shot is to protect your next child. The first child's going to be fine anyway because he's going to be born and out of there by the time the immune system makes all these antibodies. The shot is protecting the next kid. And every time this woman gets pregnant, she'll get the shot again. Because let's say she's having her second baby now. You would get the shot, coat those blood cells. Then when this baby's born and the blood enters the mom, again, it's going to be hidden from her immune system, so she won't make those D antibodies, and that's protecting child number 3. So you just keep going on. Every pregnancy, you get that shot, and it's always to protect the next child, not the current one. The current one is born and out before the immune system has time to react.